Hello students, today we will see the first lecture of BSc Third in Biotechnology and the second paper Plant Biotechnology and Plant Physiology. In the biotechnology, your syllabus focus only the technique of micropropagation in plant tissue culture. So today we will see about the introductory lecture in the plant tissue culture. So what is plant tissue culture and what is its role especially in plant biotechnology and especially in the plant breeding programs? We see that there are many techniques in plant breeding to increase the yield characteristics of our particular crop or plant. We always need the plant with special characters beyond the recombinant DNA technology. We use the different techniques of mutation breeding. We use the technique of hybridization, introduction, and so on. But plant tissue culture is especially for fast propagation of our plant. And beyond the limit of environment. How? That we will see in this lecture. You know, for growth of a plant, we need soil which contain both inorganic and organic content. Inorganic content includes some minerals. We need environment, and environment include many things like light, temperature, humidity, etc. So, but micropropagation or plant tissue culture is totally independent of these things. Means for that optimum light, for optimum temperature, for optimum humidity, we are not dependent on a particular season. हमें कोई सीजन नहीं चाहिए अपने क्रॉप को ग्रो कराने और हमें सॉइल की भी जरूरत नहीं है. मींस वी कैन सेट ए सॉइल लेस कल्चर, बट देयर आर मेनी सॉइल लेस कल्चर टेक्निक्स इन biological science, that is the hydroponics and the aeroponics and so on. What is plant tissue culture? We'll see. It is the propagation of a single cell, a tissue or organ. It is propagation of a cell, tissue or organ in sterile and synthetic food and environment. It's my own definition about plant tissue culture. मीन्स हम अपने डोनर प्लांट से उस डोनर प्लांट को जिसको हमें बहुत तेजी से प्रोपागेट कराना है, बहुत फास्ट रेट से ग्रो कराना है, या तो हम उसकी कोई एक सेल लेंगे, या उसका कोई टिश्यू लेंगे, या उसका कोई ऑर्गन लेंगे, और उसको प्रोवाइड कराएंगे क्या? फूड मीन्स मीडिया। ठीक वैसे ही जैसे कि हम माइक्रोब्स को कल्चर करते हैं मीडिया में, और वो मीडिया कैसा होना चाहिए? स्टेराइल मीन्स without contamination of another bacteria, and synthetic होना चाहिए, that should not be natural. There are some reports of natural media and plant tissue culture. Many scientists in the place of sucrose use direct the sugarcane juice, that is the. And we will provide the environment that also should be sterile and synthetic. So how we will achieve it? This cell, this tissue and organ that comes from a donor plant is called explant. And size of explant is very minute. So this technique is called micropropagation. So then first of all, we will see about food or media. That is food in our in vitro condition, because we are not dependent on external environment, and we culture the plant in a culture vessel like petri dish, that the jam bottle and the tissue culture jar. So this technique is called in vitro technique. So in the food, we use different minerals like micronutrients, macronutrients in the form of their salts like ammonium nitrate, potassium nitrate, calcium chloride, magnesium sulfate, and so on, ferrous sulfate, EDTA. We use different vitamins. We provide amino acids in that media. We use EDTA as a chelating agent. We use carbon source, especially sucrose, in that media. When we make semi-synthetic media, we also add agar in that media. Most of the cases that media we use in plant tissue culture is MS media. Murashige and Skoog media that they used, Murashige and Skoog, in 1962 to propagate the tobacco plant. That media should be sterile, means before inoculation of explant into the media, we should autoclave that media. We will autoclave that media. And we know the concentration of each macronutrient, vitamin, amino acid and sucrose in that media. So that is called synthetic media. For tissue culture, we autoclave the media because only with the help of filtering, we cannot remove totally the microbes, and the sucrose will not degrade into glucose and fructose. So our production rate will be reduced. So we should autoclave the media. We should not filter sterilize the media. Another factor is environment. So we use culture room. Like in microbiology, as we use the incubation chamber, incubation room, incubator, like that the culture room. How? We use fluorescent tubes to provide the light of different range according to our need. 1000 lux, 2000 lux light with fluorescent tube. We provide 25 plus minus 2 degrees centigrade temperature with the help of air conditioner or room heater. We provide the humidity that it needs. And culture room should be sterilized. We should not enter as such in the lab. We should use lab coat. We should sterilize our body. We use the air curtain before entering the room. So there are many methods. We use fume sterilization. We use ultraviolet light in many cases, or inoculation chamber. That is many things in the. So this was the introductory lecture and very basic concept of plant biotechnology, especially in the micropropagation. In this row, we will see another thing. This very important property of plant cell, that is called totipotency. When a plant cell has a property, that cell should be nucleated. To grow into entire plant, that cell is called totipotent cell. And each plant cell except the sieve cell of phloem has this property. And Gottlieb Haberland first of all used this term totipotency. So this is very important aspect. For tissue culture, we use this technique, and in the jam bottle, as we going to see in this diagram, first we fill the, this is not the protocol, this is only the method we are showing. First of all, we will fill the bottle with the required amount of culture media in that bottle or culture vessel. So this is culture vessel. This is media like MS media with different content. We maintain its pH and so on, that we will discuss in another lectures. And then we will inoculate explant. What is explant? From a donor plant, we use any part of that plant. That part may be apical bud, that may be axillary bud from this place, that may be part of leaf, that may be part of any root, that may be part of any nodal segment. So that part that we inoculate, the process is inoculation. Insert a cell or tissue or organ in a media is called inoculation. That small part or cell is called explant. And by using these nutrients from the media, that will grow into a entire plant within the culture vessel. And we also use different hormones for this activity. In the plant physiology, you study especially two hormones, that is the auxin and cytokinin, that we use directly in the plant tissue culture, like IAA, cytokinin, there is the BAP and kinetin. BAP is benzylamminopurine, IA is the indole acetic acid. When the ratio is of auxin is high, the result will be rooting. In the plant, rooting. And when the ratio of cytokinin will be high, then auxin, you will get shooting in your explant. So this was the introductory lecture of plant tissue culture, and with the further slides of different beautiful diagrams, we will learn about different types of cultures in your syllabus. Thank you.